Welcome to the Bible and Science. This is Dave Prentice. I'm a retired public high school teacher, certified in all branches of science and math. I've also taught on the Bible and Science for many years, both in the U.S. and on many overseas mission trips. This is episode 30 of the Bible and Science. Last few times we've been talking about could life have begun out in space and how did life begin. Now we're going to talk about how life progressed after it began. No matter what idea you have about origins, you're going to have to deal with four major areas. Number one, how did matter and energy come into existence from nothing? And number two, once they came into existence, how did they get from there to their present condition? Number three, how did life begin? And we spent a little time talking about the origin of life and about the fact that UFOs are not capable. And then once life appeared, how did it get from its beginning condition to its present state? So I want you to think about this, please. Why are you listening to a podcast called The Bible and Science? The reason I did this is I want to present indications that the Bible really is the Word of God, not just stories that people made up way back when. There may be some viewers that are undecided whether living things could be explained by chance alone or whether they point toward design and therefore a designer. So what I'm going to be covering in the next quite a few slides are intended to show those who are undecided that the features you're going to see in nature, in living things, cannot be explained by chance. They require a designer. So the question is, who is the designer? Not just some random cosmic intelligence, but the God of the Bible. I think this well illustrates the point I'm making, that you can believe in some sort of a creator that's not necessarily the God of the Bible. This particular image by Nicholas Jansen, he's been kind enough to put it out there on the internet. It intends to ridicule those of us that believe in intelligent design. He doesn't say that you have to have God. He said, well, you can have just a flying spaghetti monster. Who's the creator? Who's the intelligent designer? The flying spaghetti monster. While I disagree with that, I would say it's the God of the Bible. But this does show that intelligent design by itself is not enough. You need a personal God. So the question I want you to be thinking about is everything we see in life accidental or is it designed? Every living thing that has any structure does so because it has DNA, deoxyribonucleic acid. That's the building plan for life. DNA is a double helix structure. It's made up of millions or billions of chemicals called nucleotides, A, C, G, and T, adenine, cytosine, guanine, and thymine. They also work together in pairs, so you'll frequently hear base pairs. So DNA has millions at the very least, or in some cases, many billions of base pairs or nucleotides. So the DNA strand is subdivided into dozens of smaller units called chromosomes, which means colored bodies. Each one can contain thousands of genes. The genes are made up of thousands of those four letters A, C, G, and T, adenine, cytosine, guanine, and thymine. So by using those in many different combinations, DNA can spell out the instructions to produce any kind of living thing you can think of, and many that you can't think of. So what does DNA do? And remember, the point is this accidental or is it designed? DNA repairs the cell and maintains it. It contains the instruction to put together hundreds of copies of amino acids into each of the thousands of types of proteins the cell needs to work properly. Proteins are made out of amino acids, but it's not just random amino acids, it's specific ones that the cell needs to work. DNA also contains the instruction to link the proteins into a complete cell so it can reproduce itself, and also so that the offspring can also function properly and reproduce. The third main thing, there are many other things, but the third main thing I want to emphasize is that DNA ensures the survival and diversity of each kind. DNA contains far more genes than are visibly expressed in any individual. They're unexpressed genes, that is, you don't see any visible results, but they're there anyway. They're available to pass on to future generations. You might have heard of somebody having a throwback. They have red hair. Well, neither parent had red hair, but maybe grandpa or grandma did. Well, that's because recessive genes or unexpressed genes can be passed on to future generations. They could skip some generations. DNA can carry so much extra information that if one breeding pair had the right combination of genes, they could produce an entire kind containing multiple species and breeds. So, as I've pointed out before, let me point out again. Where did you get your DNA? From mom and dad. Where'd they get theirs? From mom and dad. Where'd they get this? From mom and dad. But you go all the way back to the beginning of humankind, and you'll find out that all of the DNA, all of the variants in the DNA, were present in Adam and Eve. And where did Eve get hers? From Adam. When God created Adam, he was already planning for you that the genes would come back together, they would recombine, and thousands of years later, here you are. God was planning you from the beginning. Now think about this. Evolutionists just overlook this. They say, your DNA came together by A, C, G, and T, and they gradually formed themselves into genes, and they formed themselves into chromosomes, and they formed themselves into longer and longer and longer structures. Well, no, this is what they're overlooking. DNA is a form of communication. It's a language written in chemicals. It specifies how to make a living thing from other chemicals. So it's communication. All known communication starts with an idea. That idea is then converted into syntax and grammar in order to be expressed. So once the syntax and grammar are expressed in words, then the idea is converted to letters and symbols to convey the message. So you start with a concept. Then you put together the grammar, then the letters. You don't just throw a bunch of random letters on the floor and hope that they'll form words that'll form sentences. No, you have an idea in mind that you want to express, and so then, okay, you convert your idea into words. All right, now how do we put the words together into something meaningful? We convert them into letters and symbols to convey a specific message. So the concept is first, then the grammar, then the letters. But evolutionists believe that the letters of A, C, G, and T evolved first and then just sort of accidentally arranged themselves into groups of three called triplets, then into genes. Then the genes accidentally came together into meaningful instructions for how to produce living things. Well, if you believe in theistic evolution, yeah, okay, you would have to say God just did all of this extra stuff. But if you're an atheist, I'm amazed at the amount of faith you have. You have the whole process backwards. But then there are quite a number of evolutionists that say, no, it's got to be natural processes only. We will not allow God in at any point. As Dr. Todd said in 1999 in Nature magazine, even if all the data point to an intelligent designer, such a hypothesis is excluded from science because it's not naturalistic. And then Dr. Watson, in the same publication, Nature, back in 1929, wrote about the theory of evolution itself as being a theory universally accepted not because it could be proved by logically coherent evidence to be true, but because the only alternative, special creation, was clearly incredible. So my mind is made up. Don't confuse me with any facts. Okay, speaking of genes, we were speaking of genes, weren't we? There are two major types of genes. There's a lot of stuff in there, but the two main ones I want to talk about. One is called homeotic genes. These are common to all members of a kind. They're responsible for the structures necessary and characteristic of the kind, very often crucial to life. So you have to have genes for hearts, lungs, eyes, and so forth. So all of the homeotic genes are expressed. They're going to produce visible results in all members of a kind. Then there can be variations, for instance, hair color, eye color, and so forth. These are genes that can exist in several different forms. They're called alleles. Some are dominant, some are recessive. Dominant genes are expressed. Recessive may not be expressed, but they can be passed on invisibly from one generation to another. Sometimes they appear as throwbacks. So grandma had alleles for red hair and nobody else did. Well, that gene may still be there and able to pop up later. Okay, every living thing known is made of cells. If anybody ever finds something that's not made of cells, that's going to be big news. The Nobel Prize Committee will no doubt give them a Nobel Prize. There are two major types of cell reproduction. One is called mitosis. The cell makes an exact duplicate of its DNA, never mind where the DNA came from in the first place, but it makes an exact duplicate. Then it splits into two daughter cells that are genetically identical to each other unless there's a copying mistake, which is called a mutation. So the vast majority of cell reproduction is by mitosis. Remember, we're trying to see, is nature, living things in nature, more likely accidental or designed? So the other sort of cell reproduction is called meiosis. Two parents each furnish a special reproductive cell with half the kind's normal amount of DNA to a single-celled offspring. Except in abnormal cases, the offspring is going to receive all of the homeotic genes from its parents. If for some reason it doesn't get them all, this is going to be a pathological condition, very abnormal. But the offspring is going to receive all of the homeotic genes from the parents, and it will also receive many of the non-crucial alleles. From the time of conception, the new organism will then grow by mitosis. So what happens if there's a copying mistake, a mutation? Unless the mutation occurs in a reproductive cell, it's not going to be passed on to offspring. So in an adult, if a mutation comes along anywhere else, it won't be passed on to offspring. It may produce things like cancerous growths and tumors and so forth. Sometimes it might not have an obvious effect. That's in adults. But in early stages of life, a mutation in an embryo or an infant will be passed on to any cells descended from the affected one. If it's in one of the crucial genes, the homeotic genes, it will almost always be harmful or perhaps even fatal. But if it's in one of the alleles of a variable gene, it will be recessive. It may not have any visible result, but it may pop up in future generations. So look at what's on the screen and tell me if you think that that is an accident. We're dealing with the question, is life accidental or is it a design? That's a model of a bovine chymotrypsin molecule from the Protein Data Bank of the Research Collaboratory for Structural Bioinformatics. But there's not just one or two different types of enzymes. There are thousands. Enzymes are special types of protein molecules. They're made of thousands of amino acids, and they serve as catalysts, speeding up a reaction to millions of times its normal rate. They have very specific shapes because of the amino acids that compose them. They have a very precise three-dimensional shape. They're so small they can capture and manipulate individual atoms or individual molecules. They have three-dimensional shapes. This allows them to snap open and closed. They're then able to either put things together or take them apart. Living things use thousands of different types of enzymes, and each kind, remember in Genesis it talks about kind, it's a broader category than species, but each kind has its own unique set of enzymes that make chemical processes happen that couldn't happen fast enough otherwise. So this is a very simple simulation when you've got a DNA strand that needs to separate because you're trying to produce a new organism. There are helicases that move along the helix structure of the DNA and unwind it. So you're seeing just a little bitty bit. But remember, the DNA can be billions of units long. So you're going to have the helix separated. First, the chemical bonds are broken by helicases, and then in order to unwind the thing, which is billions of units long in many cases, there are other enzymes called topoisomerases. These not only unwind, but they actually snip apart each half of the DNA so they won't get tangled as they're untwisting. This is just going to be impossibly hard, except that there are these little molecular machines called topoisomerases. And these are not the only two enzymes in living things. There are thousands, but this is just two that have to do with getting the process of DNA reproduction started. So the helicases break the hydrogen bonds between A and T, C and G. Study your chemistry and you'll see what hydrogen bonds are. But there's a hydrogen ion sticking out and it is going to attach to something negative. So the helicases break the hydrogen bonds between the A and the T, the C and the G. A matches with T, C matches with G. So the hydrogen bonds need to be broken on the two halves of the DNA strand, and then the helicases start to unwind the halves. Okay, once they're getting unwound, they're cut into short segments by topoisomerases. This keeps them from becoming tangled. Then there are many other enzymes, for instance, DNA primases and DNA polymerases. They use the two complementary strands as templates. These are going to be used to make two complete new DNA molecules. And then after it's done, other enzymes known as ligases rejoin the two separate single strands into the familiar structure of DNA, the double helix. So there's a model of a typical helicase enzyme. It's made of many atoms. And again, this is from the Protein Data Bank of RCSB. So after we've got the thing put back together into a single strand, we're not finished. Then it's time to start checking for errors and correcting for errors. G matches with C, A matches with T. Guanine and cytosine go together. They have 3 hydrogen bonding sites. A and T, adenine and thymine, they have 2 hydrogen bonding sites. So a G and a T will give you a mismatch. An A and a C will give you a mismatch. They need to be matched up correctly. So because of matching numbers of hydrogen bonds between pairs, enzymes copying the DNA strand usually make a copying mistake only about once in every 100,000 attempts. That's pretty good, except that the DNA can contain billions of units. So every few 100,000 is not good enough. We need to do some error correcting. There are matching number of hydrogen bonds, so A and T align at a precise distance. C and G align at a precise distance. But if any one is incorrectly paired with the wrong type, then the incorrect distances are going to produce a bump on the newly forming DNA strand. So there's a process called proofreading exonuclease. Enzymes move along the strand, lifeless chemicals that somehow knew exactly what to do. They move along the strand and they check that all of the distances are correct. There's no bumps, there's no dents. If they detect a mismatch, wow, proofreading exonucleases are going to snip out the offending nucleotide and insert the correct one in its place. So this brings the rate of copying errors down to about 1 in 10 million. So your DNA strand is reproducing, we're down to 1 in 10 million. Yeah, but that's still too much because these things can have billions of units in there. One mistake can be very, very, very dangerous, can be very deadly. So what happens now? Do we give up? No. There's another error checking mechanism, there are at least 3 of these. So step 3 is final proofreading. There's a whole family of enzymes known as DNA polymerases. They use the 2 halves of the DNA strand to make 2 copies of the original. So once the copies are complete, what you saw earlier where we split apart the DNA into its halves and then we manufacture 2 new halves, each one is intended to be identical to the original version. So after the copies are complete, some of the polymerases rescan them to proofread for errors resulting from nucleotide mismatches that somehow slipped through. So if these lifeless chemicals detect an error, one of the polymerases snips out the offending segment and manufactures and includes a corrected segment. So this reduces the rate of copying errors down to about 1 in 10 billion with a B. Human DNA has about 3 billion base pairs, so the odds are you're not going to have a copying error. Some other things might have more DNA, well they do, but the copying errors don't seem to be quite as crucial there. So all of this is done by lifeless chemicals. How do they know what to do? Is this an accident or is it something that would indicate that there's design in nature? And if there's design, oh, that would indicate a designer. So evolutionists believe that the first living things were extremely simple. DNA would not have evolved yet because there was just too much extra chemistry. So the first living things must have used some unknown storage system, far simpler than anything that's there now. So the first living things would have not had any error correcting mechanisms at all. So where did the error correcting mechanisms come from? Atheists believe these were added to DNA later, one mutation, one copying mistake at a time. So atheists have to believe that the intricate system of enzymes that prevent errors is the result of thousands of perfectly coordinated errors. That is, errors produce the error correcting and prevention mechanism. Errors produce the error prevention mechanism. Now, there are also a lot of people that are theistic evolutionists. They will say, well, yeah, I guess there's a God. They believe that God added the error correcting mechanisms much later, but then overrode them millions of times because he really, really, really wanted to have evolution. These things serve to prevent evolution. They don't enable it. So the theistic evolutionists have to say, well, yeah, God did this and then he overrode what he had just done a bunch of times, but then he put it back in place and had it start working again. I just believe that God made something that works. What I'm dealing with here is vastly oversimplified. So far, we've just talked about a primordial soup that contains carbon, hydrogen, nitrogen, and oxygen because that's most of what is in proteins. But There are also 2 amino acids that require sulfur. We have to throw that in the mix. Also, DNA and RNA require phosphorus. We've got to throw some phosphorus in there. And the early plants that were photosynthetic, would have required magnesium. And then there are many living things that also need iron, copper, calcium, and many other elements. So the soup that's proposed in laboratory experiments is just far too simple. Did you know you even contain a little bit of arsenic? There's a whole bunch of elements from the periodic table that are used in mammals such as you. So in mammals, we need a bunch of other stuff, but in plants, we need other stuff. Members of the animal kingdom sometimes need elements such as molybdenum, manganese, copper, selenium, iron, calcium, potassium, and so on, even a little bit of arsenic. So they have no way to extract these directly from the environment. It just so happens, though, that many plants furnish exactly the minerals that the animals need in a usable form. The plants send their roots out through the soil, and in a process called active transport, bring in other elements that they really are not going to make much use of themselves, but then the animals are going to need those things, so the plants concentrate them for us. So since every living thing is supposed to have come from the same single-celled ancestor sometime billions of years, there would have had to be 2 complementary and parallel series of mutations going on for hundreds of millions of years. First, animal DNA had to mutate so as to require those specific minerals. And then, secondly, since the animals couldn't get it out of the ground, the plants had to mutate to get the materials out of the ground so they would be available to the animals. I keep asking, is this accident or does it seem to be designed? Is it an amazing series of millions of coincidences, or does it point toward design and a designer instead? I hope you're noticing that we're barely scratching the surface here. People will depend upon studying biology for their careers. I'm just trying to give you a quick overview. So by all means, go stretch out your mind and look and see. I would recommend places like Answers.com, Answers in Genesis, Creation.com. A good many other places have got a lot of material. So we'll continue on with this. It seems like this would be a good stopping point. Next time we'll talk about mutations and the fact that mutations are not good things. You don't want to have them happen to you. So we'll see you then.